I spent some time up to now talking about looking at neurons and studying them in non living tissue. There's some advantages to that. We can see at the cellular level. We can't do that using current techniques. However, the main disadvantage, obviously, can't study function very easily in a non living animal. So, I'm going to tell you some techniques that we use for the living brain. These have tremendous limitations, and although they've been useful, the true use has been somewhat limited. I'm going to end up talking about CT scans, MRI, fMRI, PET. I'll mention RCBF and some other techniques. CT stands for computerized axial tomography. This is an X ray based technique. So basically, you pop your subject into the machine. The machine's going to go around and can create a 3D model of the person's brain. Now, because it's X rays, we don't get great resolution. This is what a typical CT scan looks for. If you look, here's the front, here's the back. Okay, so this would be the occipital lobe here. These are the eyes. This section is parallel to the floor. This is a horizontal section. By the way, if I'm mentioning these terms, you better know them. Okay, so this is going to be a horizontal section parallel to the floor. It's an X-ray, so you can see bone. Great. This is the skull here. You can see some fluid-filled cavities where it's black or dark. We have fluid. Places where there's tissue, we can see. So this is going to be brain tissue, but we can't see detail real well. CT scan is primarily used for things like trauma or stroke. So if I want to study the corpus callosum, this doesn't really let me see it too well. There's other techniques that are better. If a person has a behavioral deficit after a stroke and I want to figure out where the stroke is, now CT is real useful. CT also can let me see blood vessels if we inject a contrast dye. So there's lots of uses for CT scans. We're not going to talk about it too much because it's not, again, the resolution isn't great, so we can't study too much here. MRI stands for magnetic resonance imaging, based on the spin of hydrogen atoms. The machine looks very much like a CT scanner, but the image you get is different. The image you get has a lot more detail. It's good at showing soft tissues. So if you look here at this is someone who's had basically 3D imaging created using a computer of their brain. This is basically a horizontal section. This is a mid-sagittal section. A mid-sagittal section. So if you remember back, I showed you a couple lectures ago what brain looks like when it is sliced down the midline. Okay, so in this sagittal section here, you can see the corpus callosum beautifully. You can see the pons. You can see the cerebellum. You can see the medulla oblongata. You can see the sulci and the gyri. So you can see everything in the MRI that you could see in a gross dissection. Okay. Now, if we were to look at this, even on the highest power magnets, you can't see nuclei very well. There's a couple exceptions. There's some very large nuclei. Like here, for example, you can see this. That's going to be the caudate nucleus. So there's a few large nuclei that you can see. But things that we're interested in the hypothalamus, there's not enough detail here for you to see them. Okay? But what we get with an MRI is about the gross level, gross dissection. Okay, and because it's computerized, we can make all different kinds of views. Whether you prefer sagittal, horizontal, coronal, really depends on what field you're in. You'll see that For whatever reason, when you see CT scans, they like horizontal sections. When you see experimental neuroscientists like me, we like coronal sections. Others like sagittal sections. It really depends on the field. You have to get used to all three views. Right, and again, just to remind you the difference here, X-ray. This is an X-ray of a knee. This is the same structure, the knee, but this is with an MRI. So here, again, we can see much more detail. In both, you can see the femur very well. Here, though, you can see the anterior and posterior cruciate ligaments, whereas you can't see them here very well. Okay, so if we want to study soft tissue, MRI is preferable to CT scan if we're looking at structure. And again, this is an older MRI where the magnet's not quite as powerful, but you can see gray matter, you can see the white matter, you can see the corpus callosum, all those structures I talked about very well. fMRI, to make a long story short, is going to use a magnet, and it's going to look at the differences between deoxyhemoglobin and hemoglobin. So the basic idea is we measure how much hemoglobin, which is carrying oxygen, goes into an area versus how much is left at the end. Okay? And so the idea, neurons don't store energy. So a neuron that is active takes up a lot of oxygen, equals more deoxyhemoglobin. Okay. So we can use a magnetic field to detect the difference between hemoglobin and deoxyhemoglobin. More deoxyhemoglobin equals more activity. Okay, so we're not measuring activity per se. We're not measuring action potentials. We're not measuring electrical activity. What we're actually measuring is how much oxygen is taken up. Okay, and we're doing that indirectly by measuring this conversion. There's some serious concerns about fMRI, some caveats, but this is sort of the company line right now. So with fMRI, typically what they do is they take a control condition and an experimental condition. Now, at this point, if you look at any part of this brain, both of these subjects are going to be taking up oxygen. Your brain is constantly using it. So the lack of signal doesn't mean there's no activity. There is activity in both of them. Both are taking up oxygen. What you see, though, are the differences. So we'll take the activity of this and subtract it from this person. Okay? What we're left with then is a difference. So what you see in the fMRI are differences in oxygen uptake versus the control. Okay, so if neurons don't have oxygen, they die, right? So all of these neurons here, even though we don't see any coloring, those are still taking up oxygen. But it's the same amount of oxygen as in this condition. Okay? Where we see red, that means there's more activity here in this patient than there is in this patient. Okay? So you have to remember what we're seeing with fMRI are differences versus the control condition. That's why this control condition is really important. All right, and what we've got here is we've got a patient with schizophrenia and a patient without schizophrenia. And comparing, this it turns out is the medial temporal lobe. Medial frontal lobe. So if we look at the frontal lobe, frontal cortex, we see that there's more activity in this brain region in someone who's not schizophrenic than someone who is. Now, what that means is difficult to interpret. And we'll talk about that throughout the semester. PET positron emission tomography. Pay attention carefully to this, okay? Because people mix this up. It detects radioactivity. Okay? So basically, it's a machine where you put the subject in, and then there's a detector here. Anytime radiation is emitted, the detector detects it. Okay? Now, what that means depends. What does it depend on? Well, right now, if we take any of us and pop us in a PET scan, hopefully we'd see nothing. We're not normally emitting radioactivity. So, what the radioactivity is depends on which radioactive substance the patient or subject was injected with. Okay? So, all we're doing with PET is detecting radiation. That's it. Okay? Don't forget that. We're detecting radiation. What that means, our interpretation of it, depends on what we're injecting with. So I'm going to talk about one very common set of experiments that were more common before fMRI, actually. In this, we have discordant monozygotic twins. These are two 39-year-old males. One twin has been diagnosed with schizophrenia, the other doesn't. Okay. The coloring equals more radioactivity. This means less radioactivity. Okay, so what we're seeing is how much radioactivity was detected. And if you look at the arrows, you can see, if we look at the unaffected twin, they have more radioactivity in this area than the schizophrenic twin. It turns out this is, again, prefrontal cortex. Okay, so this twin has more radioactivity. What does that mean? Well, we don't have enough information yet. So I'll give you the information. We can inject with something called 2DG. That's 2-deoxyglucose. Okay, and furthermore, we can make it radioactive. So normally, neurons take up glucose to support their activity. Remember, they don't store. So they need to get it from the blood. If I increase activity, I'm going to take up more glucose. Okay. Now normally glucose gets transferred into energy. It gets broken down and gives the cell energy. 2DG looks like glucose, but it stays in the cell. Okay? So if I have radioactive 2-deoxyglucose and I give that to the patient, and then I have them do a task, any neuron involved in that task is going to increase its activity. What does that mean? It's going to take up more glucose. It's going to take up more deoxyglucose. Okay? The glucose gets converted into energy and lets the neuron do its thing. The 2DG just sits there though. Okay? So if we take someone put them in a PET scanner, then if there's a part of the brain that's working real hard, it's going to take up a lot of this 2DG. Okay, so it'll sit there and I'll see more radioactivity. So in this case, what radioactivity equals increase 2DG uptake. That's due to increased activity. Okay, so what we can conclude is there's more radioactivity, more radioactivity in the unaffected twin. It's because it took up more 2-deoxyglucose. Why do we have more 2-deoxyglucose? Because neurons in this part of the brain are more active than they are in the schizophrenic twin. Okay, so using two different techniques, we found the same result, that there's less activity in the prefrontal cortex of schizophrenics than non-schizophrenics. All right, here we've got another. We can now take a volunteer, get them in a control state, measure 2-deoxyglucose uptake, and look to see where we get radioactivity. As a control condition, they're sitting there. If you give them cocaine, look what happens. Now, this is pretty counterintuitive, right? On cocaine, you think of people being more excited, more hyper. But if you look at brain activity, it actually decreases. This is not a mistake. That's what happens. Okay, so part of what I want to point out is the relationship between 2DG uptake or neural activity and behavior is complex. If I asked, everyone would predict that this person here was more active than this person. But we know that that's not what happens. Okay, so don't necessarily interpret more activity as behavioral activation. We don't know that for sure. Okay, if we look at people who are depressed, again, we can see differences in 2DG uptake. Now remember, the PET is a technique just to look at radioactivity. What that means depends on what I'm injecting with. Keep that in mind. In all of these cases I've shown you, it's 2-deoxyglucose. All right, EEG, electroencephalogram. We'll talk a lot more about this when we talk about sleep. This studies the net activity of all neurons. It's not studying individual neurons. It's the net of many, many thousands or tens of thousands or even millions of neurons. Okay? It's the net activity of all neurons together. I can use microelectrodes where I study the activity of an individual neuron. And we've talked about microelectrodes with the squid giant axon, for example, talking about measuring action potentials. That was done in living tissue using microelectrodes. Okay? Now, this is a tough technique to learn a lot about how the brain works, right? We've got billions of neurons. I'm not going to measure from each of those. All right, microdialysis is a technique I want you to know exists. It's a way of measuring chemicals in the extracellular fluid. Basically, I just put a probe into part of the brain. So this is the brain. I put the probe. Chemicals that are released get sucked into the probe. And I can suck this fluid out and then measure amount of chemical there. So I can measure things like dopamine release using microdialysis. This is primarily non-human though. The probes are relatively large. They cause a lot of damage. They're prone to infection. This is not a technique appropriate for humans. Now, ablation has been a very useful technique. People call experimental ablation or lesion synonymously. Okay, they're the same thing basically. That's essentially, I look at behavior, destroy part of the brain, and then look for a change in behavior. And whatever changes, presumably, will reveal the function of that part of the brain I destroyed. Okay, so that's the logic behind an ablation. We do this with stroke a lot. Uh, Here, in the context I'm talking about it now, the researcher causes the damage. But with stroke, it's sort of a natural lesion. Okay, there's several different ways to make it. Here, we are going to lower a probe. So if here's my brain, I take a probe and lower a probe or electrode. Apply current and burn a hole in the brain. So if we were to do that, it would be something like that. That's the area that would get burned out. Now, when we do this, we kill cell bodies and axons. We kill glia. You kill everything in that area. It's literally burning a hole in the brain. This is the oldest technique and sort of the least refined. I can also, for excitotoxic lesions, I take my brain, I inject a needle, and I infuse something like glutamate. What that glutamate is going to do is diffuse. And it's going to overexcite neurons. This has the advantage of killing cell bodies only. It doesn't kill axons. We'll talk about chemical lesions later. These kill specific neurons. So in the case of 6-hydroxydopamine, it looks like dopamine, but it's a toxin. Any cell with a dopamine transporter will take this up and die. Right? So this is going to specifically kill dopamine neurons. All right. Now, how do we get these into the right place? Obviously, we need to get them in a precise location, or else we don't learn very much. We want to destroy a very specific part of the brain. Well, we do that using stereotaxic surgery. So stereotaxic surgery is a technique that we use in humans and non-humans to put a chemical or an electrode in a very specific part of the brain. Essentially, with stereotaxic surgery, you take the animal and you fix its skull in a consistent position in this apparatus. You can then use an atlas that's going to have X, Y, and Z axes. And it's basically a map to the brain. You just follow that. And when you use that atlas and the apparatus together, someone who's really good, looking for a relatively large nucleus, can hit 80-90% of the time. It's still very hard and it takes a lot of practice. But in non-human species, we can do pretty well. It's used in humans also. So this is specific for, here they're implanting, trying to implant fetal stem cells into the part of the brain involved in Parkinson's disease. Here they use x-rays to kind of guide them as they go. We don't do that in animals. It's not really practical. But in humans, we're going to use those. Okay, so stereotaxic surgery is just a general technique that's used to put something in a specific part of the brain. The last thing I want to talk about is connections, something about track tracing. I'm not going to go into much detail, but we can look at cell bodies. We can study cell nuclei. We can study nuclei as in clusters of cells within the cell. Within the brain. But how do we know what connects where? This is something that's really difficult to study, actually. And I just want you to know, I'm not going to go into detail about this for this course, but there's a technique called track tracing. And track tracing is going to allow us to inject chemical into one part of the brain and then see where it shows up. It doesn't diffuse, it's only carried by axons. So, for example, this FAL, we inject into a region, cell bodies take it up. Only cell bodies take it up. It's then transported anterogradely down the axon. I then cut the brain up and look to see where FAL shows up. Wherever FAL shows up, I know that the cell bodies where I made my injection send axons. Okay? So this is a tedious technique. This is not something that is used in humans. Most of the way we know what connects to where is in non-human species. Okay. So those are the basic techniques I want you to know for studying the living brain in the human and non-human species as well. These supplement what's in the book. A couple of them, like EEG, we're going to see in a lot more detail when we get to sleep and wakefulness. Thank you.